Well, welcome everyone. Welcome to our webinar and thank you for joining us. For those of you not familiar with us, Inscopix is a neuroscience tech company based in Palo Alto, California, the heart of Silicon Valley. And we provide complete miniature microscope solutions to scientists that want to map brain circuits in freely behaving animals. We're very excited to offer this Inscopix workflow webinar today. And today's speakers are Edany Hamid and Alexia Thomas. Edany joined Inscopix in 2017, and she's one of our product managers. Edany received her PhD in neuroscience from the University of Illinois at Chicago, and she conducted her postdoctoral research at NIH. Her research expertise includes EFIS with simultaneous range of imaging techniques from synaptic calcium imaging in slices to STED imaging of exocytosis and endocytosis. At Inscopix, Edany works with the Inscopix R&D and support teams to drive new product development relevant to the neuroscience market. Alexia Thomas is our application support scientist at Inscopix, and she joined in April of 2017. She received her PhD in neuroscience at Baylor College of Medicine, studying mouse behavior in an animal model of Fragile X syndrome. She continued her research studies with a postdoc position at SRI International, studying sleep and EEG in multiple rodent models of autism. Now at Inscopix, Alexia works with our customers by providing technical and scientific guidance for all Inscopix imaging projects from research design through data analysis. So before we get started, I want to make sure everyone attending understands the rules of engagement. We ask that you please provide your questions to us via the Q&A window, and we will do our best to prioritize those questions so that Edany and Alexia can answer them at the end of the presentation. Please know that we may be unable to address all of the questions depending on the time available. So without further ado, I'm going to turn this over to Edany. Okay, Edany, thanks again for joining us. I'll let you navigate the screen share from here.
Thank you, Jackie, for that warm introduction. And thank you, everyone, for joining us on our Lunch and Learn webinar series. Today's is entitled Optimize Your Miniscope Imaging Workflow. Alexia will give you the details of the surgical workflow on how to optimize your miniature microscope imaging. And we will end it with a discussion of new products and upcoming products that we're working on to further optimize the miniscope imaging workflow. Okay, but before we go into the nitty-gritty of optimization, I wanted to ensure we're all on the same page. We have here people from a range of expertise in neuroscience. So traditionally, neuroscience has been studied at the microscale, where you are able to see fine details such as dendritic spines and presynaptic terminals, but in very few neurons at a time. Or at the macro scale, we're using techniques like fMRI and PET scans, where large regions of the brain can be observed, but without the fine details that you would see of individual neurons. Inscopix was founded to address this the mesoscale challenge, enabling visualization of individual neurons simultaneously over large scale during complex cognitive or behavioral tasks. Using our technology, neuroscientists have started to map activity at the circuit and network level. At the core of the Inscopix innovation is the miniaturization of bulky benchtop fluorescence microscope into a device that can be mounted on the skull of a living animal. And this initial product was actually developed by our CEO, Kunal Ghosh, at Stanford in Mark Schnitzer's lab. You can see from this beautiful dorsal striatum recording the kind of data that you can capture using this miniature microscopes. And it's kind of currently the current miniature microscopes that we have here, just for those who know us from a lot earlier, or those that this is new to them, is Invista and Invoke. What you have here is the miniature microscope plus the black box. Using Invista, you would be able to record calcium activity. Invoke also allows you that kind of recording, basically the kind of recording you saw in the previous slide, but with Invoke now you can also manipulate calcium activity by using optogenetics. With that, I will hand it off to Alexia to go over the surgical workflow, and I will mute myself.
Okay, thank you, Adani. And hello, everyone. Let me make sure I can toggle the screen. Oop. Apologies, one moment. Okay, yes, hello, everyone. So in this section, I will be covering very briefly some of the key aspects of the imaging workflow. And I'd like to start off with a brief introduction to the standard workflow for calcium imaging. So the first step depends largely on the activity indicator you select as well as the delivery method, but typically involves optimization of a virus. This is followed by an injection of the virus in your region of interest. After about 2 weeks, a lens probe is surgically implanted in your region. We typically recommend allowing about 2 weeks for the tissue to recover before continuing to the next step, which involves installing a base plate, which serves as a docking station to attach the microscope on the animal for live imaging. Within about a week, you're ready to start collecting data with your implanted animals, and any data collected can then be processed with our analysis software. So now that you've been introduced to the standard workflow, I'll spend a little bit of time discussing each of the surgical steps involved in preparing your animals for imaging. So we're going to begin with choosing and optimizing your activity indicator. The goal of these imaging experiments is to measure dynamic neural activity. Therefore, dynamic indicators that can be expressed over long periods of time are used. The most common type of indicators currently used for imaging neural activity are calcium indicators. Here we can see the protein structure of the genetically encoded calcium indicator that the field is generally using today, GCAMP6. This is a hybrid protein that consists of a circularly permutated GFP molecule with a calcium responsive element, calmodulin, and a calmodulin binding peptide. When a neuron is active, increased levels of calcium will be detected as calcium binds, causing a conformational change and a corresponding change in the fluorescence of the GCAMP molecule, which can then be detected. So therefore, calcium is generally used as a readout of neural activity. The bottom image was acquired using a 2-photon microscope when looking at fluorescence in a GCAMP-expressing neuron. The signal-to-noise ratio is a bit better than you would expect from 1-photon imaging, but this serves to highlight how calcium imaging can be used to capture neural activity events. So there are a number of factors to consider, some of which are listed here, that are important to consider when choosing the appropriate indicator for imaging your cells of interest. Starting off with sensor types, there are a number of GCAMPs available, and it's important to consider that they each have different kinetic properties. For example, as highlighted in the table here, in particular, when you look at the last 3 indicators for GCAMP6, which come in the varieties of slow, medium, and fast, a faster indicator may mean a lower signal-to-noise ratio, which may not be ideal for your specific situation. The serotype of the virus is also important to consider, as this can affect how well the virus expresses in your region of interest. As shown in this figure, comparing 6 different serotypes, some exhibit poor expression, while others show high expression in the C1 region of the hippocampus. Selecting the appropriate promoter for your virus can also help you target your calcium indicator. The brain consists of different cell types, as we're all probably aware of, and you may be interested in imaging all of the cells, or perhaps only neurons, or maybe only glia. Therefore, selecting the appropriate promoter, like those commonly used in one-photon imaging, as shown here in the table, can help you target your calcium indicator to the correct cells in your region of interest. Other factors to consider that I'd like to just briefly mention are whether you want to consider using a Krelok system to more specifically target your indicator to a specific cell type. Furthermore, while most people use viruses to deliver their desired indicator, transgenic animals that express indicators are also available for use, which allows you to avoid using viruses. So the last several slides I showed highlight the factors to consider when choosing the indicator you want to use. But if you're using a virus, the indicator then will need to be administered to an animal by surgical injection, and therefore it's also important to consider the surgical methods used in order to optimize the expression of the virus. So just briefly, I want to highlight a few surgical considerations. Choosing the correct coordinates for the injection will be important for correct targeting of the virus. The concentration and volume of the virus, as well as how quickly it is injected, may need to be optimized. Proper expression should be enabled over the time period you will need for conducting your experiments. This type of virus optimization may require doing a series of histological analyses to confirm appropriate targeting and expression. The goal is to have sufficient expression of the virus and thus calcium indicator while avoiding under and overexpression. So in order to simplify the virus optimization, Inscopix provides functionally validated viruses for select brain regions, including the cortex, CA1 region of the hippocampus, and dorsal striatum. These viruses are ready to inject for applications in rats and/or mice, and Adenia will provide further details later in this presentation. So we just covered how to get a calcium indicator to your region of interest. Now we need a method to optically access those cells for imaging. This typically involves inserting a lens probe into the brain. Inscopix provides a number of lenses with a variety of lengths and diameters that can be used to reach more superficial regions, such as the cortex, as well as deeper regions in the brain in multiple animal models, including mice and rats. Please feel free to reach out to us for more specific details on what lenses we have to offer. So the straight lenses, as illustrated here, these enable light to pass directly from the top of the lens to the bottom. Therefore, cells located directly below the lens will be imaged. In this example movie from the Stuber lab at UNC, a straight lens was placed in the dentate gyrus of a mouse. Prism lenses, on the other hand, allow light to pass from the top of the lens down to where they exit at a perpendicular angle. This enables imaging of cells from the side of the lens. In the example shown here from the Inscopix lab, a prism lens was inserted in the cortex in order to image multiple layers of cells at the same time. So there are a couple of methods that can be used to insert the lenses in the brain. The simplest approach is a direct insertion, where the lens is gently pushed into the brain towards your region of interest. This method is often used for superficial brain regions. One concern with using this approach for deeper areas is that as you push the lens deeper and deeper, you're compressing tissue and potentially damaging the cells beneath the lens. So one method to relieve this pressure is to aspirate some of the tissue above your region of interest before inserting the lens. Please note that while I'm not going into any details of the surgical methods, we do provide surgical protocols for our Inscopix community. In terms of providing tools for optically accessing different brain regions of the brain, Inscopix has provided straight lenses and prism lenses from the time when the company started. About 5 years later, Inscopix introduced a new design for the lenses that incorporated a cuff at the top of the lens. These were our ProView lenses and were designed to be used with our ProView kit to assist in holding the lens for reliable and steady implantation in the brain. We've recently come out with a new exciting solution for lenses. These are called our integrated lenses, and Adani will be discussing these in further detail later in the presentation. Okay, so after implanting a lens in your region of interest, we now need a method to dock the microscope on top of the animal's head so that we can visualize through the implanted lens. This is accomplished by using a base plate. So traditionally, a base plate outlined here in blue, as well as in the image in the top right, is installed about 2 weeks following the lens insertion. This step involves fixing a base plate over the lens such that it's cemented to the animal's head. A microscope can then be placed into the base plate and secured with a set screw. The microscope can then be reliably attached and removed multiple times to allow longitudinal recordings of the same cells. The base plate installation procedure is a critical step in this workflow, as it must be placed in the correct orientation to allow you to visualize a plane of cells that are in focus. The focus of the microscope can be adjusted, but only within a certain range. Therefore, if the base plate is placed outside of the focusing range of the microscope, or if the base plate has been placed at an angle, there may be problems maximizing your ability to see a full plane of cells all in focus. So coming back to the standard workflow I originally presented for you, you can see that once the base plate is correctly installed, you're now ready to continue to the last steps, which are the imaging and data analysis steps. There are a number of steps that I just walked you through, and ideally, we would like to get you to imaging as quickly as possible, as that's where all the excitement is and all that data. So Adani will be presenting some of the solutions we have created to greatly streamline this whole process. And I just wanted to wrap up by kind of highlighting that even though there were a number of steps to get to the point where you are imaging and collecting data, and while this may seem a bit intimidating and complicated, we do provide a lot of resources for our community to help in this process. We provide training workshops. We have a number of resources on our online website, including protocols and background information. And we have our entire support team to help you out. And with that, I'd like to pass it back to Adani. And I am muting myself.
Thank you, Alexia, for that insightful presentation. And what I wanted to take it from there is, like Alexia mentioned, one step that we're approaching this, the streamlining of the workflow is through the support that we provide through the support team, as well as protocols that we have available. The other side we're looking at it is like, what kind of products can we develop to truly enable neuroscientists from diverse backgrounds to have access to Miniscope kind of imaging, because it truly gives a unique insight in terms of the neuroscience field. And towards that goal, we've been working on aspects of the surgical workflow to see where we can make it better. So our goals are two. One is simplify the surgical workflow. The other is how do we ensure you get reliable data faster and more easily. So I want to also stress the point about reliable data in the sense we want you to get to the imaging fast, but we want to also ensure the data you're capturing is reliable so that we truly move the neuroscience field and mental health research in a positive direction. With that in mind, I will talk about, you can already see from the schematic what Alexia was showing you was a lot longer, and we've condensed it through these new products that we're developing. So the products that I'll talk about are ready to image viruses. the ProView integrated lenses, which we really released, parts of it we released in 2019, and the full range of our lenses were released earlier this year in 2020, and the ProView Express probes. So with the ready-to-image viruses, as Alexia had mentioned earlier, the key aspect in this case is like they're concentration optimized and also functionally validated in vivo by our R&D team in Inscopix. What I mean by concentration optimized is that they've already been diluted, and for these specific brain regions we have indeed validated for, we are able to actually tell you how long you can use them, at least the part that we've validated it for. You can obviously take it longer, and you can be sure also if you use these ready-to-image viruses from animal to animal, you would have reliable data across cohorts. And so what we have here is sample data from mouse hippocampal CA1 recording, and you can see in the right the corresponding animal behavior. For this case, for example, the imaging window we're showing you is week 4, and it's you have 4 weeks of imaging available, which starts at 6 weeks from injection up to 10 weeks post-injection. Obviously, if somehow your work requires longer, worth kind of seeing how this would behave at a longer time. And below, you can see also, in this case, from rat hippocampal CA1 recordings and the corresponding animal behavior. And similarly, like the mouse hippocampal CA1 recordings, we would provide you some of the essential data that you do need to know about these viruses to enable your imaging success. So the full range of applications that at this point we're able to handle are, give me one second. For mouse brain, we have validated it for mouse neocortex, hippocampal CA1, and dorsal striatum. And for rat brain, we've validated it for prefrontal cortex and hippocampal CA1. So indeed, if these are kind of the areas that you're interested in, this is one of the steps that would indeed move you faster into imaging. On the other hand, of course, if you also have your own optimized virus, that's actually also another option. So the other one I wanted to talk about is the ProView integrated lens. So with the ProView integrated lens, it's the product we announced in the last SFN, which feels right now a bit of a lifetime away. But at the key aspect to define it is base plate with attached Grin lenses. And if you look at the overall step, you can see in this case, the only part you need is virus injection, then implantation. The implantation basically combines both the lens implantation and base plating. So the key benefit is, again, like I stated, is increase your efficiency by reducing the number of surgical procedures. It's one surgery instead of 2, which was normally lens implantation and base plating, but now into one surgery together. And also, it shortens your time from viral expression to imaging. If we go back to Alexia's talk, she had mentioned, for example, after base plating, typically you would wait about a week before you image. This would be something that would be shaved off because this step is done together. The next aspect is the other really key benefit here is that we optimize the working distance for in vivo imaging. And I'll go through that one a bit more. As Alexia mentioned, for example, base plating becomes a key step because while quite often we just purely say it's a docking station, but it's really also a point in the surgical workflow where you determine the optical path. So in a typical and standard workflow, you would have Grin lens implanted first, and then your base plating. So we generally can give you the work distance of each of our lenses, and we have a range for each of the lenses. However, where this range will end up lying would truly depend on where you base plate. So like Alexia mentioned, if you're completely out of the range, even though the microscope indeed has like 300 micrometers of range in Z, you could be completely out of the range. So what we've done with the integrated lenses, this was really a combination work with the in vivo R&D team, as well as the engineering team and myself, to come up with what would be the ideal precise working distance range for in vivo research. And we were able to achieve that. And indeed, with the integrated lens, not only are you saving steps in surgery, we're basically now providing your product where the optical path from the lens all the way to the microscope is completely idealized. So in terms of going back to kind of bring it back to the workflow, with this product, one, we're streamlining the surgical workflow, but also removing a component that does indeed require skills and making it something that's already pre-done for you to ensure your success is, again, much faster. And you get to the point, like Alexia said, where you're seeing cells, you can actually analyze and make insightful findings out of those. And if you have more questions about the integrated lenses, so if you're part of the Inscopix community, reach out to support@inscopix.com. If you're new to us or are already talking to some of our sales managers, feel free to also reach out to them directly or the sales@inscopix.com. It is a product we really literally released in March, actually, so a little bit before the shutdown started. So the next one I wanted to talk about is the ProView Express probe. So the ProView Express probe actually preceded in terms of our release of it from the ProView Integrated Lens. It was released in 2019. And specifically in this case, the product is a virus-coated lens plus base plate, which enables you successful imaging with a single surgery. And you can look at it here. If you look here, even with the ProView Integrated Lens, for example, you do have 2 steps. And now with the ProView Express probe, we're getting it to a single step. And if we especially compare this to the traditional process, which was up to about 5 steps from optimization, injection, lens implantation, base plating, we're now into 1 step. So we'll reiterate it's a single step surgery. But second, other aspects that's really key to this is that the GCAMP virus expression is localized to the lens surface. So thereby enabling you to boost the number of cells you will image in the position you're implanting. The other part that's really key is you are able to obtain reproducible imaging results over time and across cohorts. This is enabled by the fact that, like the RTIs, we do actually optimize the concentration of virus that's painted on the probes. Of course, what's always exciting about a lot of these recordings is the beautiful imaging data that you can get. In this case, so for our ProView Express probe, it's been validated in the mouse dorsal striatum, and you can see here the vector. It's an AAV1 with a CAMK2A promoter, so it is more general. It expresses both in D1 and D2 medium spiny neurons in the dorsal striatum, and we always give you the window of imaging. If you can image at the minimum for 4 weeks, you can start imaging from the 3rd week all the way to the 7th week post-implantation. You can see on the left the recording, in this case, from about 6 weeks post-implantation and the corresponding animal behaving here. And what we have in these last 2 panels here is post at the end of like these validation stage. If we do histology, you can see the lens track here. And if we look at the magnified component here, this is a histology that has DAPI staining, as well as you see the GCAMP in green here. But we can kind of see what's really neat about the Express probes is you end up really labeling just about 150, 200 microns of the area right below the lens. And what's been like kind of a positive beneficial effect to this is that you end up reducing your expression in the areas that are out of focus, which ends up revealing a better signal to noise ratio. And in this case, because it has been out a little longer now, it was out in 2019, it has been tested by labs outside of our own lab. And when you can see that this is data from external labs, you can see they're imaging at week 3, week 5, week 7 post-implantation, and you can see from the different labs here. And the key benefit here is consistency is quite easier. And in the cases that you do follow the full procedure and guideline that you would get with this product, the success rate is quite high, both for novice users and for expert users generally. So I wanted kind of to end our last section of our talk to be about products that are coming soon. It would be more exciting to talk about what is coming soon. And one of the things we have been working on is another ProView Express probe. As soon as we released the earlier ProView Express probe, the common question was, can we release it in a version that is actually more specific, that is enabled to label specific types of cells? So with that in mind, the new virus that we are currently validating and hope to release in about 2 months to 4 months is a cell type specific virus coded lens, but plus base plate, but again, to enable you successful imaging. In this case though, in the dorsal striatum in D1 or D2 medium spiny neurons through a single step surgery. You can see the viral vector here. It's an AAV-DJ. It's a CAG flex. So with the flex, and when you combine that with a Cree mouse line, you would be able to get that specificity. The exact lens type is a 1x4 millimeter integrated lens. And we again provide you the imaging window, which is 4 weeks. Start imaging at week 4 all the way to 8 post-implantation. Our validation, as I said, is ongoing. What we're working on is using D1 and D2 Cree lines. The ones we're using specifically are DRD1 Cree mouse line and Adora 2A Cree mouse line. These are one of the more commonly used Cree mouse lines for specifically labeling D1 and D2 medium spiny neurons. And here to show you some of the data from our R&D team. This is the work of Dave Chang, and he does make some beautiful recordings of the dorsal striatum. This is D2 medium spiny neuron imaging 6 weeks post-implantation with one of these ProView Express probes. And on the right, you have the corresponding mouse behavior. And overall, whenever we do any of these RTIs or the ProView Express probe, we also give you the important numerical data that you would be interested in these kind of cases, like the kinetics, the number of cells, reach rate, or event rate as well. And you can see that listed here and below. As I mentioned, of course, we're validating it not just in D2 medium spiny neurons, but in D1 medium spiny neurons. And this is sample data from D1 medium spiny neurons imaged 4 weeks post-implantation. And again, here on the left, you see the recording of the dorsal striatum. And on the right, you have the mouse behaving. In this case, it's kind of a bit cool in the sense you can see when the mice pauses, you see a reduction in the number of activity. When the mice is moving, you see an increase in activity. I'll admit I'm not an expert in movement neuroscience or dorsal striatum, and I'm sure this network activity would mean a lot more to those scientists. Like the other data, we do provide you the cell number and kinetics and overall other information that you would be most interested in to ensure your work also is consistent and you can actually make some sense out of the kind of data you're getting out of these express probes. Of course, when talking about selectivity, you want to ensure, are they truly selective for the specific cells you're going for? So towards that end, we're confirming selectivity. To confirm that, we're crossing our, for example, in this case, D1 Cree mouse with AI14 mice. AI14 mice are reporter mice. When crossed with a Cree, the TD tomato would express in that situation. So what we've done in this case is, like with these crossed mice, we inject the full concentration of our virus. And 4 weeks after survival, we sacrifice and do histology on them. The counts that we're showing you here is from 3 separate regions of interest per animal. And what we're doing is we're looking at GCAMP expressing neurons and to see how many of them were co-labeled or not with TD tomato. And what we find is overall 96% of the cells that are expressing GCAMP also express TD tomato. And if you look at this data, what you see here is the data that you see in the left here in green, the data of the TD tomato here in red, and they're just flipping back and forth for you to be able to see, like, what cells are indeed like showing up in both cases. With that, I kind of wanted to end it a bit more to show you what has been the evolution of the Miniscope workflow improvement. We started off in 2011 with the miniature microscope being developed, and then 2012 being commercialized. Along with the miniature microscope, the Glyn lenses were also released, because that is the way you are enabled to access deeper brain regions than beyond the few layers of the cortex. And in 2016, like Alexia had mentioned, the timeline, we would release the ProView lenses. The ProView lenses indeed made a huge difference in the sense the surgical steps were improved quite dramatically from the naked Glyn lenses to the ProView lenses. In this sense, the success rate went up for a lot of labs quite dramatically. And now in 2019 and 2020, what we're releasing is not just improvement of the surgical workflow, but we're going to the point where we're optimizing your whole optical path and also truly ensuring your reliability goes up. So it's kind of like not just success, but also reliability of the data that you're getting is going up through these new developments that we have. I know Alexia and I really focused on optimization of the workflow, but what's really exciting, I mean, optimization of the workflow can be exciting, but really what's cool about this research is the kind of neuroscience information that we're gaining from them. And towards that end, we are doing bi-weekly Insights webinar series. Our latest one will be next Wednesday at 11 a.m. Pacific time with Dr. Mooney from Duke University. I urge you to join us for one of those, and also worth seeing even some of the previous recordings for the last two Insight webinar series we've had. Another place that is worth visiting is the publications page. We actually list the full publications that are coming out using INVISTA and Invoke. And at this point, we've gotten over 120 peer-reviewed publications. So worth really looking at to see what kind of research insight is being developed. And of course, if you have more questions or just want to discuss some of the science, please reach out to support@inscopix.com and the sales@inscopix.com. All right.
Thank you, Adani and Alexia. This was a great presentation. I want to remind everyone who's attending this webinar, you're welcome to send us some questions via the chat box. And I'm sure either one, Adani or Alexia, would be happy to answer any questions you might have. I think the workflow improvements that we have demonstrated today could really benefit all of the future circuit neuroscience researchers in trying to collect what can be sometimes complex behavior, as well as understanding those circuits that mediate that behavior. Wouldn't you agree, Adani?
I just muted myself. Yeah, absolutely. In terms of, because one of the things actually which I should have mentioned, for example, with the Provi Express probe, we have a lab. One of our users, for example, are studying Huntington, and the model animal they're using dies around 8 weeks post-birth. So you kind of are left in a position where without the Provi Express probe, that work can't really be done at all, because if you go the normal route of viral injection and then lens implantation and everything, it's really past the point where they can image. But with these Provi Express probe, they're actually starting to work where they implant, and 3 weeks post that stage, they start actually doing some of these recordings. So we do really see a huge benefit from these different workflow improvement. And then with the integrated lenses, where I can see that is, like Alexia mentioned, base plating is indeed an important step. And with that step, we basically remove that step through this development. You're getting something that's already optimized. As long as you do your lens implantation properly and use our guidance, I would say our support team is always still useful when making sure you implant in the proper manner. You should really improve your success rate quite dramatically.
Thank you, Adani. Yeah, I was also going to just ask if the integrated lenses, where everyone has, most scientists likely have their own research interests in a very particular brain region or a field of study. These lenses are actually offered for all lengths, correct?
Yes, yeah, they are. We're very excited about that, because when we released it in 2019, it was really only the 1x4 millimeter lens, which is one of the most commonly used for, let's say, dorsal striatum, hippocampus, to name a few. But we now have released it for all our lenses. When I mean all, it's like 9 lenses. So you can kind of also imagine the R&D. Like I said, it's not simply gluing lens with base plate. It's optimizing the working distance for all of these lenses. So we now do have them available for everything. but also the prism lenses, just to be clear.
And given their point on the prism lenses, we did receive a question from one of our attendees. Can you give us a feel for what that working distance from a prism lens might be?
Yeah. In terms of that, the working distance actually for the prism lens is quite large. As we were doing this R&D, I think we discovered a bit more about all the lenses, and you would be able to get from zero meaning if this is the, like, let's go to the lens. Give me a second. If we go to one of the, I'll go to Alexia's slide actually on prism lenses. So in general, what we've optimized it is to be where you would be able to get image any cell that is at zero from the tip of the lens all the way to approximately around 400 micrometer. I might be off on the exact number of the actual range, but for that kind of detail, always worth actually asking, like talking to our sales team or our support team so that they can send you a spec sheet. But yeah, the range is quite a bit. But again, when we're talking about the prism, we are talking on this side, right? When we're talking about the straight lens, we're talking about from this point of the lens all the way down. But from this case, it's from here to here. So the lens range is actually a lot more than even the microscope range. But just when talking about imaging in the brain, because the brain is quite a bit of a fatty tissue and light scatters, in the sense, so going beyond 300 micrometer or even 300 micrometer is not always essential, basically. So you would be able to get exactly what you need with the prism lens.
And perhaps a reminder with our systems, the ability to image through the tissue is one of the benefits, I believe, is the electronic focus, correct?
It is, yeah. And the electronic focus, it's two sides to it. One is once you've done your lens implantation, again, we're talking about obviously you want to optimize, make sure these things are well done. And the second part though is the full range is 300 micrometers. So as long as when you've implanted, you are within that range, you can go back and forth. And then other aspect of the electronic focus that I really think is worth really remembering is longitudinal recordings, because if you want to do longitudinal recordings, which is really key benefit for a lot of the insights we're gaining from these kind of recordings, is the eFocus. Just basically all you have to do is save that data in our software, and when you're imaging from say an animal, you just go back to it, and you'll be able to. Another key point, just reminder, for all these things, especially like the lens implantation, we provide protocols and guidelines, and you have access to neuroscientists like Alexia, who has extensive in vivo research experience to truly guide you on how to do it. Because indeed, it might seem simple, right? It's a lens, just shove it in and you'll get what you need. But it's not that. Like Alexia said, you don't want to depress too much to the extent what you're imaging is not really what you think you're imaging. So you want to use these people. And Alexia, would you want to add to that a bit more? A bit more to to that? In terms of the electronic really focus.
I think, yeah, well, I think the only other thing I can think of really to mention, because I think if people are familiar with some of our older microscopes, the eFocus is just an easier thing to work with, because even when we're talking about longitudinal recordings, I mean, sometimes, things change over time. I mean, we're talking about biology. And so, it's possible that you get slight shifts in tissue around the lens over time. And so even though Danny talked about the ease in being able to save your settings so that you can easily return to those settings for a specific animal over multiple days of recordings. But if you need to do any fine-tune adjustments to make sure that you're right at that crisp plane to see those cells over multiple days of recordings, the ability to make those fine adjustments is very simple.
Thanks, Alexia. And I'm just curious, can we provide a more descriptive way of how we would look at electronic focus or the digital focus through our software for those that may not be familiar? Maybe a Danny?
Well, yeah.
Or yeah. Do you want to?
Sure. Yeah. So if you look at in our software, Inscopix Data Acquisition Software, or IDAS, what you'll see, the lens focus is the UI you see there is from 0 to 1,000. That actually linearly extrapolates to what is for our focus for the microscope is 50 microns up to 350 microns. But when we're talking about the lens itself, that will, when we're doing in vivo imaging, that translates to 0 being 0 and all the way to 1,000 being 300 microns, basically. So it's a linear relationship.
Oh, that makes a little more sense. Thank you. And I think perhaps, Alexia, coming back to the support side, we have numerous guidelines and protocols, correct?
Yes. And we have protocols that are more general, so that, you know, everybody's looking at different brain regions, they have different approaches. So we try to generalize some of our procedures, but then if people, you know, a lot of people, let's say, we have some like some areas that a lot of people focus on, like striatum, hippocampus. So we have some brain-specific protocols to help with that.
Yeah, thank you. Thank you. Well, I want to thank everyone for their participation, and I certainly want to thank you both for doing this with us. This has been a really informative webinar. I hope everyone who attended today found this information useful and interesting. I would ask all of you to stay tuned for future webinar dates. We have a lot of exciting virtual events planned in the coming weeks. And again, please feel free to reach out to us on our contact page at Inscopix. And I hope you all have a wonderful rest of your day. Thanks again, everyone. I'm going to go ahead and end this webinar. Have a great afternoon. Bye.